Hi students, I am Professor Nanasaheb Gunjal from Sri Sai Baba Senior College Shirdi, Department of Botany. Due to the interim vacation of coronavirus, there is need to interact and communicate with the students online. That's why I am going to start my first video tutorial lecture on the mitosis. First of all, let's start with the basic concept of cell division. A cell division is the process by which a cell divides into two new daughter cells. But what is the need of cell division to the organism? The answer is all the living things want to grow and develop by producing number of cells by means of cell division. If there is damaged tissue in the organism, then there is need to repair it by means of cell division. As well as if cell gets too big, it cannot get enough nutrients, then there is a need to be a divide cells. Okay, this is the actual process of cell division which is explained here. The original cell is called as the parent cell and divided cells are called as the daughter cells. This is the parent cell, and after division there are two daughter cells which are produced by means of mitosis. Before cell division occurs, the cell replicates its DNA. So each daughter cell gets the complete set of genetic information, that is chromosomes, from the parent cell. Each daughter cell is exactly like the parent cell and it has the same kind and the number of chromosomes as like the original parent cell. The many organisms, especially the unicellular organisms which are reproduced by means of cell division, that is called as the asexual reproduction by the binary fission. The example is bacteria. Before going to start the process of mitosis, we should know the basic details and the concept about the nucleus, chromosomes, chromatin material and the DNA. So, the DNA is located inside the nucleus, inside the chromosome, which is made up of chromatin material. Chromatin material is consist of the DNA and histone protein. The chromosome looks like X shape in condensed form of chromatin. If chromatin is in relaxed state, then there is only looking like as a thin thread like mixed nuclear structure. Chromosome has centromere and the chromatids when it is in condensed form. Now this is very important part about the chromosome. Always students going to misunderstand about these two structures, that is the unduplicated structure of chromosome and the duplicated structure of chromosome. When two identical sister chromatids are attached at the area in the middle, that is called as the centromere, then the structure is formed, that is called as the duplicated chromosome. Each duplicated chromosome consists of two identical sister chromatids having same sequence of genes on the chromosome. When cell divides, the sister chromatid separates from each other in the anaphase and each cell gets one chromatid, and this chromatid is nothing but the unduplicated chromosome. Here is the chromatin to chromosome illustration, that is how chromatin is condensed into the chromosome, how relaxed state chromatin is get condensed, and what is the need to do this to the cell. In first figure the chromatin is unduplicated, after which it gets duplicated in synthetic phase of the cell cycle, and after that there is a need to condense it in the third figure. The question is that why does DNA need to change from chromatin to chromosome? In another language, why does DNA need to change from relaxed state chromatin to the condensed state chromatin, that is the chromosome? The answer is more efficient cell division, because cell want to divide. If cell want to divide, it should divide its unduplicated chromatin into the duplicated form, and after duplication there is need to coil it, there is need to package it inside the nucleus. In this slide we are going to discuss the variety of chromosome number among the organisms. Each and every organism has its specific number of chromosomes inside their haploid and diploid cells. The first example is the human. Human has 46 chromosomes in their diploid cells, and in their haploid cell there are only 23 chromosomes. 23 pairs of chromosomes are divided into two parts, that is 22 pairs of autosomes and one pair of sex chromosomes, that is X and Y. Dog has 78 chromosomes in their diploid cell, and in their haploid cell there are 39 chromosomes. Goldfish has 94 chromosomes in their diploid cell and 47 chromosomes inside the haploid cell. Lettuce, 18 chromosomes in their diploid cells and 9 chromosomes in their haploid. Here we are going to discuss the chromosome number in the various somatic cells of the organism. Take the first example of human being. Human being consists of 46 chromosomes in their somatic cells. In skin cell as well as in heart cell as well as in the muscle cells, there are complete total 46 chromosomes in their somatic body cells. The second example of the fruit fly. The skin of fruit fly and the heart cell of the fruit fly consist of 8 chromosomes. Also in muscle cells of fruit fly there are 8. From number of slides we are discussing about the mitosis, but this is actually the start of this mitosis process. The cell cycle is a series of events cells goes through as they grow and divide. So in cell cycle there are number of events successively arranged inside the growth and development. So as the cell grows, it prepares for division by accumulating nutrients, after which it divides to form two daughter cells, each of which then begins the cell cycle. The cell cycle has two main phases, that is the interphase. Interphase is nothing but the growth and development phase or preparatory phase for the mitosis. In the interphase, cells start their growth by accumulating nutrients and synthesizing some proteins or molecules. Inside the cell, there is a need to replicate the DNA, to make copies of DNA for the mitosis. So in interphase, cell going to grow and it will carries all normal cell activities and replication of all other organelles. That's why cell requires maximum time of their life cycle for the interphase. The second phase of cell cycle is called as the mitosis. Mitosis is the division of nucleus into two nuclei and each cell with the same number of chromosomes. Actually, the mitosis is divided into the two parts. The first part is called as the karyokinesis and second part is called as the cytokinesis. The karyokinesis is nothing but the division of nucleus. Karyon means nucleus and kinesis means division. So karyokinesis, and second one is the cytokinesis. Cytokinesis is nothing but the division of cytoplasm. The question is that where mitosis is occur? The answer is mitosis occur in all the somatic cells of the plants and animals. So what is the need of mitosis? Why does mitosis occur? The reason and the answer is each new daughter cell has nucleus with complete sets of chromosomes. So each daughter cell gets its cytoplasm and the nuclear or a genetic material by means of cell division, by means of mitosis, which is explained in the below images. In this slide, we are going to discuss the karyokinesis. Karyokinesis is the first part of the mitosis. In the karyokinesis, there are 4 stages: prophase, metaphase, anaphase, and telophase. The first stage is called as the prophase. In prophase, the chromatin material is thin thread-like. The nuclear membrane starts to disappear by degradation. Nucleolus also starts to disappear in this phase. The centrosomes are divided into the 2 parts that makes the centrioles, and these centrioles are moving from each other. The second stage is called as the metaphase. In metaphase, centrioles are completely moved towards the opposite poles of the cells. Nuclear membrane completely disappeared. Then there is a condensed form of chromatin in the form of condensed chromosome. All the chromosomes are lie at the equator. Each chromosome is bounded by the spindle fibers oriented from the 2 opposite centriole poles. The third stage is the anaphase. In anaphase, there is a separation of sister chromatids by using the spindle fibers. Spindle pulls the sister chromatids from each other and get separated in the anaphase. The last stage of karyokinesis is the telophase. In telophase, there is a starting of formation of nuclear membrane, and there is a starting of formation of cell plate for the division furrow. So we have to identify the various stages of the mitosis. The first stage is prophase. In prophase, chromosomes coils up, that is starts to condense. Nuclear envelope starts to disappear. Spindle fibers starts to form due to their centriole division of centrosome. The second stage, identify this. This is the metaphase. In metaphase, chromosomes are lined up in the middle of the cell, that is at the equator. Then second point, that is the spindle fibers connects to the chromosomes centromere. This is the third stage. Identify it. This is the anaphase. Anaphase consists of separation of chromatids, that is chromosomes copies get divided. The spindle fibers pulls the chromosomes, that is pulls the chromatid to the opposite poles. Identify this. This is the telophase. In telophase, chromosomes starts to uncoil, that is they are going to make their thin chromatin again. Then nuclear envelopes start to form. The two nuclei are formed inside this. The spindle fibers disappears, that is centrioles or centrosomes are disappears. The second part of the mitosis is called as the cytokinesis. Cytokinesis is the division of rest of the cell cytoplasm and cell organelles after the nuclear division, that is the karyokinesis. In animal cells, the cytoplasm is pinches in after the telophase. In plant cell, the cell plates are formed after telophase. This is the basic difference. All students have to understand this important point, that is, in animal cells, after the telophase, cytoplasm is get pinched inside. Okay, and in plant cell there is a formation of cell plate, that is the cell wall formation is there. After the mitosis and cytokinesis, the cell returns to the interphase to continue the growth and development and perform all the regular cell activities. Look, there are some activities about the how cleavage furrow is formed in the cytokinesis. Here, all these explained here in some figures. Let's start the summary of cell cycle. Interphase and the M-phase. Interphase is the preparatory phase where cell accumulates nutrients and synthesizing some proteins and divides the DNA. After which mitosis starts. Mitosis consists of karyokinesis and cytokinesis. Karyokinesis consists of prophase, metaphase, anaphase and telophase. And after the telophase there is a cytokinesis, that is the division of cytoplasm and some cell organelles. When cells become old or damaged, they die and replaced with new cells. This is the main function of cell cycle. Here are some actual images taken by some research workers and scientists to demonstrate how cell cycle works. So students have to observe all the details about the cell cycle and its division stages. Actual and demonstrative images are very different, but we have to complete it in your practical. So it is very important to understand all these real images. So it is very important chart and table about understanding the various stages of the cell cycle, having the interphase and the mitosis. So we have to remember all the things by doing own study, your own readings, by drawing some figures and listening this lecture. So I hope this lecture will helps you in your study of your interim vacation. Thank you. So thanks. This was my first lecture of the mitosis. Due to the coronavirus interim vacation, there is a need to interact by this online video tutorial lecture. We are making number of videos for the students to understand the number of various concepts. So, thank you. Thank you.